I like to start by celebrating the beauty and diversity of plant cells, as shown in this slide. These are some stunning images of plant cells generated by members of our community. I am but one of almost 50 core members of the Plant Cell Atlas community, most of whom are early career scientists, ranging from grad students to postdocs to assistant faculty level folks. And it is an international community, not only of public and private universities and colleges, but also including nonprofit research groups, as well as government research centers. So we care deeply about the looming climate crisis and the associated issues with food security, environmental protection, and energy production. And we believe that plant science has a huge role to play in our future economy and society. Just as an example, I like to share with you that scientists project that biomass will become the global primary energy source in the next 30 years. The graph on the left, I don't know if I can, okay, there you go. The graph on the left shows you the amount of primary energy supplied by these major sources, such as biomass, wind and solar, coal, gas, and nuclear. The black bars represent the amount projected in 2020, and the blue bars represent the amount projected to be produced by 2030, and the red bars represent the amount projected to be produced by 2050. And as you can see, biomass will become the biggest source of energy globally in about 30 years. However, this comes with a huge caveat in that biomass is a big energy hog. As you can see on the graph on the right, the amount of greenhouse gas produced as a function of land intensity or land use, biomass, even though it produces a lot less greenhouse gas, is going to be using the most amount of land compared to things like coal and natural gas. So a big challenge that faces us in the next 20 years or so is really the problem of how do we get the most out of plants, such as energy, food, and even medicine, while minimizing resources and reducing environmental impact. For this, I believe that we need to really be able to develop a programmable plant that we can manipulate at will. In order to do this, we really need to understand major processes of plants, such as development, reproduction, metabolism, defense, and physiology, and so on. Not only do we need to know how they work at molecular details, we also need to understand how they work together or against each other, what the trade-offs are between these major processes. I think there are a couple of major bottlenecks that we need to overcome in order to achieve this ambitious goal in a relatively short amount of time. I think one of the biggest roadblocks is that plant science is still quite underrepresented in STEM. This graph shows you the number of soil and plant scientists working in the US today relative to the number of medical scientists taken from the Department of Labor of the US. And you can see that the number of soil and plant scientists represent only about 10% of the number in medical scientists. And so we need to figure out a way of recruiting a lot of talent into the plant field. The second major hurdle that I see is that of all of the genomes that have been sequenced so far, a huge portion of them are still encode genes that have no known function. So each pie chart here represents the genome, the number of protein encoding genes of various organisms. And the green pieces of the pies represent those protein encoding genes that are experimentally characterized. Blue pieces of the pie represent the genes whose functions can be predicted based on their similarity in sequence to other characterized genes. And finally, the black pieces of the pie represent the protein encoding genes that are completely unknown for function, and they're not even, they don't even look like anything that's been characterized. And so how we can figure out what these proteins and genes do I believe represent a huge bottleneck that we have to overcome, not only in plants, but in any biological field. So how can we really accelerate the discovery here, especially in plant science? To answer that question, I like to use this analogy of a jigsaw puzzle. When I do a jigsaw puzzle, which I enjoy doing a lot, in the beginning, it's really hard. It's really hard to know where to put these pieces because they're really out of context. But as soon as I start to be able to put together a few of the pieces into their relevant context, then all of a sudden it becomes really easy to put the rest of the pieces together, and the process of solving the puzzle really accelerates. I think we can take this analogy to biology and really think about the location to function paradigm, which states that the location of molecules are essential in identifying the functions of their functions. And this paradigm, I think, can be extended at multiple scales, ranging from molecular to subcellular to cellular, organ and organism levels, and perhaps even at the ecosystem level. And it is this paradigm that we'd like to establish in the plant cell atlas community. And so a couple of years ago, a few of us got together to try to create a community effort to map all of the components of plant cells from nano to macro scale resolutions. We understood that clearly this is not something that can be done by a single lab or even a few labs. And so we started building this community, and our major aspirations are as follows. So our goals are to first identify cell types, cell states, and transitions between the states and plants, different plants, and to map proteins to different cell types and subcellular structures, as well as to track the dynamic interactions among the proteins, and finally to integrate these data to generate multi-scale models of cells and tissues. We hope that the work from this community will help answer fundamental questions in plant science and also accelerate engineering of plants for agriculture, bioenergy industries, and ecosystem management. Finally, we'd like to use this community effort as a vehicle to engage the general public about plant science. The research framework that we're envisioning is as follows. We think that this is going to be a very intimate collaborations between plant scientists, people that are developing technologies, engineers, and so on, and data scientists who are really interested in integrating the data. In this process, we think that data standardization will become really important, both for manual and automatic ways of curating and analyzing data. So some of you might wonder what kind of science this PCA effort will enable. Well, I think that if done successfully, the data and the know-hows and the best practices will impact all areas of plant biology, some of which are described in these small balls here, and lots of major areas of biology that are currently kind of difficult to prove will become much easier, and we will be able to get to a deeper level of understanding. For the lack of time, I won't go into all of these different areas. But I will just give you one example of an area of biology that I think could really benefit from Plant Cell Atlas Initiative, and that is C4 photosynthesis. So photosynthesis, as some of you might know, is able to sequester a huge amount of carbon dioxide every year. In fact, it can do about 200 gigatons of carbon dioxide per year, which is more than 10 times, almost 20 times of the human generated carbon dioxide per year. Of course, most of these carbon that's fixed is returned as a function of respiration by the plants. But the point is that plants actually are capable of fixing huge amount of carbon dioxide. And what's even cooler is that plants that can do C4 photosynthesis, which is a specialized type of photosynthesis, can actually fix about a quarter of the CO2 in the world. And there are only about 3% of flowering plants that do C4 photosynthesis. So you can see already from these two numbers that C4 photosynthesis is really an amazing invention that evolved in plants. And just to very briefly describe to you how C4 photosynthesis works, I'm going to start with introducing C3 photosynthesis in a very high level. So this is a typical cross-section of a leaf that you can see a few different cell types. The most of the cell types here are called mesophyll cells that are the factories that do the photosynthesis. And then you have this vascular tissue, which you can think of as arteries and veins of plants that basically transport water and mineral nutrients as well as the sugars that are fixed. And then these vascular structures are surrounded by what's called bundle sheath cells. And I'm not going to go into the biochemistry too much, but the basics of photosynthesis is that carbon dioxide is captured into sugar by this enzyme called ribisco in a pathway called Calvin cycle. And the energy comes from light reactions carried out by photosystems 1 and 2 that generate ATP and NADPH. And then the byproduct of this light capture and ATP and NADPH generation is, of course, the splitting of water to generate oxygen. And so really the poop of photosynthesis is really the oxygen that humans and other organisms on Earth depend on living. Now, C4 photosynthesis is really cool in that it specializes this really a beautiful biochemistry into two cell types. And it really separates the carbon fixation from, carbon capture from carbon fixation and the light energy capture into two cell types. And so the mesophyll cells basically become this structure called Cran's anatomy that surrounds the bundle sheath cells and the vascular structure. And the mesophyll cells in C4 plants are focused on capturing the light and generating the energy like ATP and NADPH. And it created a new pathway called C4 pathway, or the carbon concentration pump pathway, that captures CO2 and then puts it into a 4-carbon sugar. And that 4-carbon sugar is then transported into the bundle sheath cells. That's kind of hidden away from the rest of the tissues. And this is where then the carbon dioxide is released from the 4-carbon sugar and then fixed into other sugars and biomass and so on. And the reason why plants invented this is because this rubisco that takes carbon dioxide and fixes it into sugar is not very good at distinguishing between carbon dioxide and oxygen. And so when you actually have conditions that reduce the amount of carbon dioxide or increase oxygen, like dryness or increase in temperature, you get a lot of this reaction with oxygen instead of carbon dioxide. So it's a really nice way of concentrating carbon dioxide that plants invented using these sort of cellular structures. So I think this is a really fascinating area of plant biology, and we still don't really know how all these different cell types function and how they develop and how they react to different environmental conditions. And so being able to probe this question at the individual cell type level, at the bundle sheath and the mesophyll cells, would be really powerful to elucidate how C4 photosynthesis works. So then I'm going to start wrapping up my talk by describing some of the challenges and opportunities that we see for the PCA. As alluded to by Ed in the previous talk, in the plant field, we really need to think about how to integrate single cell analysis across different species, and how to really create these maps of different organs and how those different organs and cell types will react to different changes in environments. And so, just because we're at the very early stage of building this initiative and getting into single cell technologies in plant sciences, we have a lot of challenges and opportunities ahead of us. So the way we're thinking about addressing these opportunities and challenges is really to continue the grassroots activity of building the community. And so of the 50 or so people that I mentioned in the beginning of this talk have self-organized into about 14 committees that range from different aspects of science, such as single cell, and technologies such as single cell sequencing, proteomics, and imaging, and so on, as well as infrastructure related to committees such as data management, data infrastructure, outreach, communication, and so on. And I'm really glad to see that these committees are led by an amazing group of energetic and enthusiastic and charismatic early career scientists. And I want to do a special shout out to Selena Rice Carnegie, who is the coordinator for the PCA community and has been doing really the lion's share of the work behind the scenes. And finally, this PCA community is supported by a smaller group of not so young scientists, but very ambitious and service oriented at the same time. So together in the last couple of years, we've achieved some major milestones. We had a vision paper published in Trends in Plant Science a couple of years ago. We obtained a grant from NSF to do a first workshop. We launched a website and Twitter. We held our first workshop virtually last spring. And then we have a special call for papers for a focus issue that will come out at the end of this year in plant physiology. And the report from our inaugural workshop has been published in Plant Direct. We launched a YouTube channel earlier this year. We did another workshop virtually on single cell sequencing. In February, we did another one on spatial proteomics in April. We obtained a research collaboration network from NSF, which starts June 2021. It will go on for the next 5 years. We're submitting an application for a Gordon Conference to start in 2023. Our roadmap paper is actually under revision currently, and hopefully will be published this year. We are overhauling our website and launching the Generation 2 website this fall. And we're actually developing a science and art exhibit about plant cells at 3 historically Black colleges and universities that will start this fall. And then we're going to do another workshop on mass spec imaging in the fall. And then finally, in December, we're planning on a virtual international PCA symposium. And currently, we have about 790 members in our group, about 500 followers on Twitter, just starting to get some subscribers on YouTube. This beautiful diagram generated by Maite Saura Sanchez represents the stakeholders that we envision. We would love to get more connected with members of the industry. And if you are wondering how you might be able to get involved, these are 5 different ways you can get involved. You can find out more about our efforts on this website, plantcellatlas.org. You can join our email group by just putting your email and contact information on this website, plantcellatlas.org/contact-html. You can follow us on Twitter at cellatlas. You can also subscribe to our YouTube channel. And finally, feel free to email us at plantcellatlas@gmail.com.